Thank you very much, Professor O'Doherty. Can I please ask Professor Pääbo to come forward now and deliver his keynote address?
Clap afterwards. Mr. President, dear members of the King family, dear organizers of the conference, dear friends, first of all, let me then thank you for the honor to be at this fascinating conference and for the honor to speak here tonight. So what I then wanted to do, and please tell me in the back if you don't hear me, because I can't quite judge how loud I am. But what I wanted to do then is describe to you our efforts to sequence the genome of the Neanderthal and of some relatives of Neanderthals, other extinct forms of humans. And then in the end of the talk, the last third or so, discuss a little where this field might go in the future. But before then doing that, I wanted to remind you about what you are all well aware of, that our genetic material, our genome, is stored in almost all cells in our body, on the chromosomes, in the form of this famous double helix, the DNA molecule, and it's stored in the form of a sequence of these 4 bases, and it's stored twice, so to say, one on each strand, because whenever you have one base, an A here, you have a T on the other, and a G there, you have a C on the other strand. And this is very important because when a cell divides, particularly then when cells in our germline divides, these strands come apart and you synthesize new strands with the old strand as a template. So you then end up with 2 copies which are essentially identical. But nothing is, of course, perfect in nature, so sometimes errors are made, say the wrong letter here is built in, and if that's not repaired fast enough, it will appear, if it happens in the germline, in the next generation as a mutation. And we detect those mutations when we compare genomes between 2 people today, for example. We will find a difference approximately every 1,000 such letters or so differ. And in every new baby that is born has something like between 50 and 150 new mutations that's not there neither in the father nor in the mother. So these mutations rain down on our genome approximately as a function of time. So if we now add to these 2 humans, add a chimpanzee genome, we will find a difference approximately every 100 letters. So you can then reconstruct the history of a piece of DNA by looking at those differences you observe here, reconstruct what has happened in the past. And in this case, in a very simple tree depict that. The 2 human sequences go back to a common ancestor quite recently, and about 10 times further back is a common ancestor shared with the chimp also. So our genome, as you know, is about 3 billion such letters long. So it's quite a lot of information there, around 3 million differences or so between 2 individuals' genomes you compare. So it's a great source of information to reconstruct our past. So if you do that on a worldwide scale, what you will find is that you have the most variation among people in Africa. And if you look on the entire world outside Africa, although much more people live outside Africa than inside Africa, of course, you have less variation there. And not only that, the genetic variants you find out here generally have close relatives inside Africa. But there is then, in addition, a component in the genetic variation in Africa that you don't find outside. And this is sort of the genetic background for the recent origin of humans, the hypothesis of a recent origin of humans in Africa, suggesting that modern humans originated in Africa, accumulated genetic variation there, and then went out, a part of that genetic variation, so to say, went out and colonized the rest of the world. And with some genetic tricks, you can even estimate approximately when this happened, and that's in the order of 100,000 years ago or so. But there is a problem with this recent African origin idea about modern humans, if you like, and that is that at that time, say 100,000, 50,000 years ago, modern humans were not alone on the scene. There were other forms of humans around, most famously then Neanderthals in Europe and Western Asia, and in East Asia, other forms that are less well defined. So a big question since a long time is sort of how do they relate to present-day people? So these Neanderthals, as you well know now, both from the introduction a little while ago and from the films we saw before that, are these robust forms of humans that then appear in the fossil record in the order of 400,000 years ago until they become extinct around 30,000 years ago or so. And there are then two ideas around in paleontology since a long time about what happened when modern humans came out of Africa, come to Europe and Asia. One idea is that you replace the Neanderthals with no mixture whatsoever. There's no contribution from Neanderthals to people today. And another one is an assimilation, or that modern humans come out of Africa, replace these forms, but there is a contribution. So Neanderthals today would be on average closer to people in Europe, and other forms in Asia closer to people in Asia. So if you like, these two ideas can be seen as a continuum to a total continuity that no one really believes in, that the Neanderthals are the direct ancestors of Europeans, to total replacement, zero contribution from Neanderthals to people today. And we got the first chance to test these ideas genetically back in the 80s when Ralph Schmitz, who's here in the audience, together arranged for us to be able to try to study DNA from the type specimen. So this is then not just any Neanderthal, it is actually the type specimen found in 1856 in Neanderthal. And it is also then the specimen that William King used to name this species 150 years ago. And I think we were actually very lucky to be able to work on the type specimen first, because it's also the fact that whatever Neanderthal we since have studied, there's always some paleontologist who come and tell us it's not quite a typical Neanderthal. It's a little too gracile, it's a little robust, but if this is not a Neanderthal, Neanderthals don't exist, so to say. So Ralph removed a sample from the humerus here from us. At the time, we used quite a bit of material, much less these days. You extract the DNA under clean room conditions to avoid contamination from your own DNA into the experiments. We, at the time, with the technology of the time, focused on a small part of the genome located outside the nuclear genome, the mitochondrial genome that's inherited from the female side, on a particular variable part of that, cumbersomely retrieving many short pieces, replicating the experiments many times, believing the changes that were there in all our experiments, and estimated such a phylogenetic tree, as I showed you before. And what that showed was that the mitochondrial genomes and this part of the genome for present-day humans go back to common ancestor around 100, 200,000 years ago, and much, much further back, about 500,000 years ago or a bit more, is the common ancestor with the mitochondrial genome of Neanderthals. Since then, we and others have studied other mitochondrial genomes from other Neanderthals. They all fall together outside the variation of present-day humans. So in this scheme of things, then, it was quite clear. There is no person today running around with a Neanderthal mitochondrial genome, so there is total replacement, 0% contribution. So this work also gave us another insight, and that is this approximate time here of 500,000 years as a time point where after which the population split between Neanderthals and modern humans must have happened. So that is quite recent in human evolution. It's also quite recent from a genetic point of view, because we know that if we compare two genomes between two people in this room, we go back to a common ancestor on average somewhere around 500,000 years ago. And there's a big variance around that. So you can easily find some part of my genome where I differ from someone in the audience by 1,000,000 years. So for a substantial part of our genome, our variation is actually older than the split to the Neanderthals. So that means that without studying more of the Neanderthal genome, we could immediately say they must, in the nuclear genome, often fall within our variation, simply because we're so closely related to them. So for some part of the genome, someone here will be closer to Neanderthal than some other person in the room. And in another part of the genome, someone else will be closer to Neanderthal. But that doesn't mean then that having a Neanderthal genome would be uninteresting, because there will still then be things, parts of the genome, that will have changes that have happened recently, since we separated from Neanderthals, that make us unique relative to Neanderthals. And I think I'm on the public record somewhere like 10 years ago or 9 years ago of saying we will never have a Neanderthal genome. It's too degraded, it's too little DNA there, it can't be done. Of course, you should never say things like that, especially not in science, because you're generally proven wrong. And it's generally technology that ends up proving you wrong. And in this case, it was clear it was high-throughput DNA sequencing that changed the whole scheme. The ability that came in the early 2000s to sequence large amounts of DNA sequences rapidly and inexpensively. So what you can then do is simply extract the DNA from such a fossil and not try to focus on any particular part of the genome you're interested in, but just sequence randomly all the DNA molecules that are in there. Make yourself a little database and compare it to the human genome and find the things that come from something human-like that you then believe is from the Neanderthal. So the first place where this worked was Vindhya Cave in Croatia in southern Europe. And this little bone, which is around 38,000 years old, or this little piece of this bone. And the first thing that you then notice when you study the DNA in this is that it's indeed very short pieces. So whereas a blood sample from me, you would get DNA fragments that are 10,000, 20,000 such letters long. Here it's an average of 40, 50 letters. The other thing you will notice is that just a tiny minority of the DNA in the bones come from the Neanderthal. The vast majority comes from bacteria and fungi that colonized the bone after its death. So the very best Neanderthal bones is something like 3 or 4% that we have found. So we were very lucky to have funding over a 4-year or 5-year period to try to work on retrieving a Neanderthal genome. So we got a lot better in the methods we use to count from the bones to something you can feed into the machines, and the machines got better. And we looked through a lot of Neanderthal sites and many bones, and in the end focused on 3 bones from this Vindhya cave in Croatia, from 3 different individuals that we used together to sequence a bit over 1 billion DNA fragments, that at the time seemed to be a lot. The vast majority then from bacteria and fungi. And you then, with computer programs that we partly had to develop for this and had to puzzle this together, moving these short damaged fragments and aligning them with the human genome. And in 2010 then we had about a little over half of the genome that we had seen at least once, sometimes more times. So we could begin to we had an overview for the first time of the genome and could begin to ask questions. And one of the first questions was then this: what happened when Neanderthals, when modern humans came out of Africa and met Neanderthals in Western Eurasia? Did one mix or not? So we approached that question in 3 different independent ways. I just want to present one here, the most intuitive one, maybe, that if there was a contribution from Neanderthals to Europeans, we would expect Europeans to share more genetic variants with Neanderthals than Africans do today, because there have never been Neanderthals in Africa, so we would not expect a contribution from Neanderthals to Africans. We are then looking, so it's this idea again: if there is no contribution, the Neanderthal is equally far from someone in Africa as someone in Europe. If there is a contribution, on average, the Neanderthal will be closer to Europeans than to Africans. So we were then looking for small signals, potentially small signals in the genome. So we were very worried about differences in errors in the DNA sequences we had that other labs had done. Not that other labs sequence less well than us, but there can be differences in the types of errors when different labs do it. So we decided to go out and sequence 5 genomes ourselves from present-day people. So we sequenced these 5 individuals, 1 European individual. To us, of course, the archetypical European is a French person, so we sequenced a French person. 2 African individuals here, 1 person from China and 1 from Papua New Guinea. And then did a very simple analysis, comparing 2 different present-day humans, and we start with 2 Africans, where we expect no Neanderthal contribution. We simply find all places where those 2 genomes differ, and then we take the Neanderthal and say, how often does the Neanderthal match that African or that African? We expect that to be 50-50, right? There's no reason for one African to be closer to a Neanderthal than another African. And indeed, that's the case. Statistically speaking, it's about 100,000 matches to each of these genomes. So if we then go on and do this for the European individual and the African individual, now, to our surprise, I must say, we found statistically significantly more matching to the European than to the African. That surprised me. I was sort of biased to think there would be no contribution from Neanderthals. But if there would be one, I would expect it in Europe. So it was even more surprising that when we compared a Chinese person to an African individual, we again found more matching. And even more so in Papua New Guinea, it was surprising that we saw it there again. So the sort of hypothesis we suggested to explain this finding that was surprising, that we see the contributions also in parts of the world where Neanderthals had not existed, was to say that when modern humans come out of Africa, they presumably passed by the Middle East. And we know there lived Neanderthals there. So if one then mixed with Neanderthals in the Middle East, and if these modern humans went on to become the ancestors of everybody outside Africa, they would sort of carry with them this Neanderthal contribution, also to parts of the world where Neanderthals had never been. To the extent then that our best estimate today is somewhere between 1% and 2% of the genomes of people, all people outside Africa, come from Neanderthals. So something else have also appeared recently. Several groups have realized that there's actually even slightly more Neanderthal DNA in people in Asia than in Europe, which is surprising. And recently in January, Josh Akius, also at this meeting, have shown that the best explanation for that is that one actually has at least 1 more contribution of Neanderthals to people in Asia after this contribution that presumably were to the ancestors of everybody outside Africa. So we then have at least 1 more case of mixture with Neanderthals that add something like 20% extra, so from 1% to 1.2%. This is worrying. It says battery is running low here, 10%. I hope this works. So we then published this in 2010. And there were a lot of scientific follow-up work of this, which is very satisfying. But we also very soon sort of noticed that the public was also quite interested in it. We started receiving a lot of letters and mail, and I can never stop myself from mentioning this, from members of the public that sort of self-identified as Neanderthals. And after a while, I started sort of seeing a pattern in this correspondence that surprised me. And that was that, I will do this instead. That was that it was mostly men who wrote to me and self-identified as Neanderthals, and very few women. So I sort of presented that to my students as my research. I count emails. And they then said, this is just ascertainment. It's just that men are more interested in molecular genetics, so they will write to you. Females will not do that. But I went back to my mails and found that, my letters, and found that that's not true, because there were actually quite a few women who did write to me and said they were married to Neanderthals. Whereas so far there's not a single man who is married to Neanderthals. So this, as a geneticist, you of course get very interested in such inheritance patterns. But it of course represents sort of our social ideas about what Neanderthals are like, that perhaps also partially go back to William King again, who I quote here as saying that in the skull, the thoughts and the desires that were there never soared beyond those of the brute. So maybe that's something that sometimes can be seen as attractive in a man, but rarely in a woman. So that's probably reflected in this correspondence. So another important question is, of course, when this contribution happened. And there are a number of different genetic ways to get at that. One way developed by David Reich's group that we collaborate with at Harvard is to say that if we had these two populations, Neanderthals and modern humans, and they mixed with each other, the first hybrid individual will of course have one chromosome in each pair from a Neanderthal, one from a modern human. If these individuals now continue to live with modern humans and have children with them, you will then have recombination, where in each generation chromosome pairs exchange pieces here. So these pieces that came from the Neanderthals will in each generation get smaller and smaller. Initially they will be quite big, and with time they become a lot smaller. So what you can then do is to look at the size of these pieces in the genome that come from Neanderthals and try to date when they entered the modern human gene pool by their size. So look for things like this. This is a part on chromosome 21 where the Neanderthal genome is very similar to some Europeans but quite different from everyone else. So this is very likely to come from a Neanderthal. So what they did was to use this and do a number of assumptions about how this recombination between chromosomes happen and generation times and so on, and arrived to a date between 40 and 90,000 years ago approximately for this. But this relies on a lot of models. And I guess if there's some sort of principle in my work is in a way that when you rely on models, you never really know if your models are good enough. So I would love to actually go back and ask when this happened. And it begins to become possible now. So there was a bone found 3 years ago now at a river close to a place called Oceania in Siberia, at this place. It's a humerus, and it seems to be a modern human. And when we got this to our institute in Leipzig and carbon dated it, we were amazed to find that it was 45,000 years old. So it's a very early modern human. It's actually the earliest directly dated modern human outside the Middle East and Africa. So a question is then, does this individual have a Neanderthal contribution or not? Because it falls in this range, of course, when it could have happened. So if we now look just for chromosome 12, I should say we've sequenced this genome of this individual now to high quality. Just look on one chromosome in some present-day humans and mark here in the Neanderthal pieces that one chromosome comes from the Neanderthal in red and where it's homozygous in green. And we can differentiate here actually between the Europeans up here and Asians, and you can sort of squint and even see that Asians seems to have a bit more contribution from Neanderthals, as we have said. But the question is this now, this 45,000-year person up here, do they have a contribution from Neanderthals or not? And indeed, it turns out, oops, sorry, I'm bad at this. They do. But as you can see, they have much bigger chunks, which is of course exactly what you would expect if it happened rather recently. They haven't been degraded to so small pieces yet. So you can sort of use association of Neanderthal-like variants in the genome in present-day humans, as we just discussed, to arrive at this date of 40 to 90,000 years ago. This is this 45,000-year-old individual, and that estimate is somewhere around 300 generations before this individual lived, 45,000 years ago. So we arrive at a date somewhere 50 to 60,000 years before present for this contribution, under the assumption that this happened in one event, which is really an oversimplification, but it gives you a feel for when this can have happened. So what else are we interested in? We're clearly interested in other forms of extinct humans other than Neanderthals. And we're very lucky to work with colleagues in Russia, particularly Professor Derevyanko in Novosibirsk, who excavates at many different sites in Siberia, particularly this site here, the Denisova cave on the border to China and Mongolia. This site is a beautiful area where they in 2008 found a tiny little bone that they were actually very skilled to realize it could come from a human. It's a part of the last phalanx of a pinky of a child. So we analyzed this bone, the DNA in it, and we were amazed to find that it was very well preserved. As I said, our best Neanderthal bones have something like 4% endogenous DNA. This has 70%. So using then methods that we recently developed in the last few years that are much more sensitive than earlier methods in retrieving small amounts of DNA, particularly because we actually separated two strands, and each strand is then made to come into the library. So each double-stranded molecule has two chances, if you like, to make it into what we sequence. So we're able to sequence this genome to very high quality. So each position is sequenced approximately 30 times over from 30 different DNA fragments. So every position in the genome to which you can map these short fragments is seen here. So what we then found was that this individual in the Denisova cave has a common origin with Neanderthals here, but Neanderthals since have a long independent history. So sort of realized we have to name this other group something, and just like Neanderthals are then called Neanderthal after the first place where they were found, we call these guys Denisovans since they were first found in the Denisova cave. So we now have a high-quality genome for the first time of an extinct relative of Neanderthals, and we actually now have a Neanderthal genome of similar quality. So what you can then do is that you can actually distinguish the two variants that this individual got from its mother and its father in the chromosome pair. And what you can then do is estimate the time back to a common ancestor between these two samples of two genomes in the population this individual came from, coming back here. And you can then go across the genome and do this for many positions in the genome and get the distribution of times back to a common ancestor between these two genomes that this individual carries. And of course, in the past, if the population was small, you will more often come to a common ancestor when the population was big. So in this case here, if you infer the population size back over time, what you will see is that you will have a sort of inferred dip in population size here, where you have many of these things go back to common ancestors. So you can then go and do this for present-day humans first, and you will see that no matter where you live today, in the past we share a reduction in population size, an increase in population size, and only quite recently, in the last 100,000 years ago or so, do we begin to see difference between Africa and non-Africa, where this reflects then a migration out of Africa more recently. So we can now do this for this extinct form of humans, for these Denisovans, and trace their history, starting in the past. And you will see that if anything, they're slightly bigger in size here, and then crash down and become extinct. So quite fascinatingly, this individual comes from a population that has a history that is very different from anybody that lives today. Something else that you can do when you now have high-quality genome from extinct individuals is that you can start to see that this individual lived in the past. So it's sort of missing mutations because it lived a number of thousands of years ago when you compare it to present-day humans. So if you do that here, you can see that counting back to the common ancestor with the chimp, it misses something like 1 to 1.3% of mutations. If you assume for the moment that this was 6.5 million years ago, this means that this individual lived 60 to 80,000 years ago. So it's quite fascinating to me that from a bone that's so small that you cannot do carbon dating of it actually, when we can get the genome, we will in the future at least be able to approximately date it. But I should say there's a lot of caveats at the moment, for example, due to the differences in qualities of present-day genomes. But this is certainly something that will become more and more possible when we start sequencing more accurately in the future. So it's fascinating to me that from a very tiny bone like this, you can actually infer a lot about the population history, the age of the bone, but frustratingly, we don't know the morphology of these individuals, we don't know what stone tools they made, and so on. But it's something I think we will see more of in archaeology in the future. We can ask for these Denisovans also, just as for the Neanderthals, did they contribute to present-day humans? And indeed they did, but surprisingly not in Siberia, but rather out in Oceania here, to the extent that something like up to 5% of the genomes of people in Papua New Guinea or in Aboriginal Australians come from these Denisovans. So if we should summarize before coming to the last part here, our ideas from studying genomes then about the origin of Neanderthals and modern humans. We believe that Neanderthals and Denisovans have an origin in Africa in the order of 500,000 years ago or so. They come out of Africa, presumably mix and then evolve in Western Eurasia to what we call Neanderthals, and in Eastern Eurasia to what we would call Denisovans. This is not to say that Denisovans were ever this widespread or that there weren't other forms of hominins there. We actually don't know where the border between them were at any one time. We do know that in this part in Siberia, at some time there were Neanderthals, some other times Denisovans. Then modern humans have an origin in Africa, come out of Africa, start spreading seriously around 50, 60,000 years ago, mix with Neanderthals, presumably in the Middle East, continue to spread out across Eurasia, mix at least once more with Neanderthals, mix with Denisovans further to the east, in presumably Southeast Asia somewhere, and continue out into the Pacific. And these other forms of humans then become extinct with time, but live on, if you like, then a little bit in the genomes of present-day people here, and up to a total of 7% or so in Oceania. So what we clearly have done is sort of refute this total replacement scenario here. We see up to 7% or so of the genomes of some people come from these extinct forms. So somewhere we have picked up this sort of term of leaky replacement. The big picture is still replacement, but there is some leakage, if you like, of genetic material from these other forms. There are, of course, others who have suggested similar models in the past. For example, replacement with hybridization, to Breuer, or assimilation by Fred Smith. And I have no sort of stake in what we call these things. So what has then happened recently is that Anatoly Derevyanko have again found interesting things in Denisova cave. Another small bone, this time a toe bone, deeper down, this bone. When we looked at the genome from that one, we found to our surprise that it was close to Neanderthals and not to this Denisovan. So we now have a very high quality Neanderthal genome also. And again, you can begin to look at things such as variation between the two chromosomes. What you find is that you look at present-day people, you have more variation in Africa, less sort of diversity outside Africa, and these extinct forms have even less variation. And we started seeing another interesting pattern too, that if we looked at this Neanderthal individual, we have long stretches where the two chromosomes inherited from the mother and from the father are identical to each other. Much less so than what we see in the Denisovan individual. This, of course, suggests that the mother and father of this individual were closely related to each other. So we can then sort of estimate what relationships could have been. It has to have been one of these four ones we have there. So the parents of this individuals were either half-sibs, say grandfather, granddaughter, and don't even ask me what the double first cousin is, I have no clue, but it's depicted there. But they were clearly closely related anyway. So it will be very interesting to see when you get high-quality genomes from other Neanderthals in other parts of the world, if this was a general pattern or something that went on just in this cave. So we then now have these two high-quality genomes here, a Denisovan Neanderthal. We have a lower quality genome from the Caucasus that I didn't mention, and this from Croatia here. So we can now begin to look at contribution not only to present-day people, but also among these extinct forms with each other. So if we do this, we can look at modern humans, Neanderthals, Denisovans. We see this contribution from Neanderthals to modern humans that we already know outside Africa. We see this contribution that we also know from Denisovans into people in Melanesia. What we now also see is a small but discernible contribution from Denisovans also to people in mainland Asia, in China, for example. We also see a contribution from Neanderthals, particularly these eastern Neanderthals, to Denisovans, a rather small contribution. And quite interesting, an old component in the Denisova genome that we don't see with the Neanderthals, that comes from someone else. A hominin that diverged earlier along the lineage to humans into here. Presumably, it's hard to estimate how big that contribution is. It's a few percent at least. And the divergence here is also hard to estimate, but in the order of 1 to 4 million years ago or so. So it's very tempting to suggest that this could be Homo erectus or something like that, that mixed in Asia with Denisovan ancestors. So a take-home message from this is that we have always mixed with each other. And at least a little bit, we find no mixtures that are really very, very big. It's generally a few percent. And I'd like to end then by bringing up a few things where this might go in the future. 3 small things. One, I think, is actually to go further back in time, till Neanderthal ancestors, for example. So we can use these hypersensitive techniques now to go back to older fossils. And the place where we've been able to do that is a place in Spain, Cima de los Esos, with these presumably Homo heidelbergensis that are 400,000 years old, so 5 to 10 times older than the Neanderthals we looked at so far. It's a deep cave site where they found many bones, and from this femur here, we were able to extract a little bit of DNA. And just to give you a feeling, from 500 million of DNA sequences, we sort of filter it down and end up having 10,000 fragments approximately that we can use to look at this mitochondrial genome. And quite surprisingly there then, it turns out to be related, this mitochondrial genome, not to the Neanderthals, but far back, but still with Denisovans. So these forms that we found over in Siberia. So it's very unclear why that is the case. Perhaps we're simply so far back in time that we're back in sort of close to common ancestor between Neanderthals and Denisovans, or maybe this comes from someone else, from Homo erectus or so, also into these individuals here. But the fascinating thing is that we begin to be able to go further back in time. I'd always said, say, 100,000 is somewhere at the border of this, but now we can clearly go back 400,000 and perhaps approaching 1 million years or so in the future. Another thing that will come is, of course, and the beginning to come, is looking for a functional contribution from Neanderthals to present-day humans. Have they influenced our biology today? There begins to come work from that, again from Josh Akey's group and from David Rice's group, where you can look, for example, in Europeans and find that generally, if you look across the genome, it's very low percent of people who have contributions. But there are some things that have shot up to high frequency where 50–60% of Europeans carry something from Neanderthals here. Some things are shared between Asia and Europe, some are Asian specific. So if you just ask statistically what types of genes are in those parts that reach high frequency, the thing that comes up are keratins, so structure proteins in hair and skin. So there might be that there's something in the hair or skin of present-day people that do come over from Neanderthals. Another thing that fascinated me is that late last year was a group in the US who found a risk variant for type 2 diabetes, so the type of diabetes you get in old age. And this is a lipid transporter with several differences to the non-risk alleles, and it occurs particularly in East Asia, a little bit in Europe, not at all in Africa, and also in Native Americans. And when you compare this risk allele here to non-risk alleles in a tree, you find that the Neanderthal version falls right in with the risk alleles here. So this version have come over from Neanderthals to present-day people, risen to high frequency in Asia, perhaps because it may give some advantage in starvation or something like that. And today, when we have ample nutrition, it sort of causes diabetes. So I think we will see more and more of this in the next few years, that we actually start understanding how these archaic forms actually contributed to us today. You can, of course, also look across the genome, the different chromosomes here, and look for parts of the genome where we have no contribution from Neanderthals, although we would expect to see it statistically. So there are regions like that indicated here. And you can ask, what are those regions where we seem to resist Neanderthal contribution, where we seem to not accept it and select it away? And if you just ask what genes are there, where are those genes expressed in our body, it turns out that the only place, statistically speaking, they are enriched is genes that are turned on in the testicles, in the male germline. So that makes it very tempting to suggest that there was something problem with fertility, with male fertility in the hybrids. So that's of course a common phenomenon. If a horse and a donkey has offspring, the male offspring is actually infertile and the females are fertile. So it may be that there was actually some sort of incompatibility between these groups when they mated with each other. And finally then, what I think actually is almost most fascinating is to look for what functional things are unique to modern humans. So now saying, when we have the high quality genome from our closest evolutionary relatives, now we can focus on these things here that changed here and became fixed in all humans. No matter where we live on the planet, we all carry these changes, but at those positions, the Neanderthal look like the apes. The things that happened here, and if you like, then define us as a species or as a group relative to our closest relatives. And that's a sort of potentially interesting set of mutations, because of course some things that happen with modern humans, technology starts changing very rapidly, start having figurative art that actually depicts things, and we start spreading all across the world, coming to every habitable place that exists on the planet. So we can now make this catalog, and the fascinating thing is that this catalog is not very long. So if we take a very strict view here, say we should, as far as we know, have these things in 100% of people today, Neanderthals and Denisovans look like the apes, then that's a list of 31,000 changes in the genome, approximately. So that's not very much. You can look through it in an afternoon. So for example, if you just think about amino acid changes in proteins, the molecules that actually does jobs in our bodies, it's 96 amino acid changes in 87 genes. So now we come though to the problem with the genomics, and that is a dirty little secret of genomics, that we are incredibly bad at, from any particular mutation we see, sort of say what function that change has in the genome. But of course, we would try, so we can see where these genes are expressed, for example, because tend to think that things active in the brain might be particularly interesting. So I was quite interested when some collaborators of us actually showed that in the developing human brain, it was particularly in the part of the brain where the cerebral cortex is formed from precursor cells that these genes are expressed. And there are 5 genes expressed there. These are small numbers, of course. It's in total only 87 proteins we studied. But 3 of them turned out to be involved in cell division, how the chromosomes are separated in cell division. So that was very surprising to me, that something so basic would differ between modern humans and Neanderthals. But there are, of course, indications that how these stem cells divide when the brain is formed. The cleavage plane, for example, determines what types of neurons you form and how many of them you will form. So it may be that these 3 genes, for example, involved in cell division here in the brain might be particularly interesting to look at. But then it comes to the very last thing I wanted to bring up, and that is a question, how should we study these now in the future? How should we study human-specific genetic changes and Neanderthal-specific changes? Because we don't really have an animal model, of course. These are human-specific. So I've gone around for the last 10 years and say that obviously what we want to do is to make a transgenic human where we put in the Neanderthal versions, and transgenic chimps, so we put in the human versions, but we have ethics committees and things that make things difficult for us. Well, that's not only a joke, it's a sort of how will we all come forward, go forward with this. And it's also not so much a joke, because there are scientists who have started suggesting that we should consider at least the possibility of cloning Neanderthals from stem cells, which is of course an even more extreme idea than this. But fortunately, that will also not be possible. But what can we do in the next 10 years or so? Well, I think one thing that will happen is that when everybody who goes to the doctor has their whole genome sequenced, just as default, and that will come rather soon, I think, or within the next 10 years, then we know that there are so many people on the planet and the genome is so small that every mutation compatible with human life exists there in the population. The genome is 3 billion bases, we have 7 billion humans, every baby that's born has 100 new mutations, so everything exists there as a mutation, but again at very low frequencies. But we will then be able to find back mutations, if you like, to the ancestral state and study them, I think. But what we can also, of course, do is engineer these changes, if we don't want to wait for this, into stem cells and in vitro, in the tissue culture, study their effects. And after all, also put in these changes in genes in mice, and humanize mice and Neanderthal mice, and look for their effects. So to end then, I hope that I've convinced you that if you're interested in human evolution, recent human evolution, it's very useful to have our closest relative, the genome, our closest relative. You can see what has happened here, and you can also see what has happened there. We haven't dwelled on that, but when we have more genomes here, we will also see what has happened there. But that will not be enough. You will need functional tests, and one will then be stem cells in tissue culture, but another, I think, important one will also be humanized mice. And with that, I then thank you for your attention. I cannot mention everyone who was involved, many people in Leipzig, many groups that contributed fossils to this, many people that helped analyze this. I should mention particularly Monte Slatkin and his group in Berkeley, and David Reich and his group at Harvard. I will mention one person, and that is Matthias Meyer in the lab, who developed this hypersensitive new way of making libraries from single DNA strands. Without that method, we would not have had the high coverage genomes from Neanderthals and Denisovans. And with that, I then thank you for your attention.
Professor Pääbo, that was absolutely amazing. Thank you very much. Would you like to take some questions from the audience?
I'd be glad to take questions.
Yes. Would you like some water, maybe? Whilst we'll take some questions from the audience. We have 2 roving microphones, so if you'd like to ask a question, please feel free, but please wait for the microphone to come to you. So, who wants to go first? So over here.
Thanks for a really amazing talk. I was just wondering, you didn't mention Homo floresiensis from the Flores Island. Is there any hope of retrieving DNA from any of those specimens?
I would never totally give out a hope, but Yeah, it was 7 years ago, I think, we were allowed to drill in the root of a tooth a little bit, much less than we would have liked to have, and couldn't retrieve any DNA. Now we are better today. One should perhaps try again, but it's humid and hot on Flores. It's sort of bad conditions. I would be pessimistic actually about that. It would be very interesting, of course.
Just down the front here.
I'll just talk loudly, will I? Thank you. Thanks very much for the talk, it was fascinating. I'm just trying to envisage what it was like in Europe, let's say, when Neanderthals and Homo sapiens existed together. Can you estimate, from your percentage contributions that you mentioned there, what sort of size of population was interbreeding with itself, as it were, or with each other? What numbers are we talking about?
Well, I sort of certainly can't do that. There are other people, perhaps Josh is better at estimating those things. I have gotten a little skeptical about that when we have now, about estimating that, when we actually tend to find that selection is involved in some of this. We see some variants of Neanderthal contributions that risen to high frequency. We see other rather big parts of the genome where we seem to resist it. So if it was a total random process, I think we could model it quite well. Now we would have to take in sort of an estimation of what selection is going on and in what directions. I'm sure there will be smart theoreticians that will come and approach these things, but it will not be so easy, I think.
There's a question over here.
Hi, thanks for a great talk, it's very interesting. I just wondered, is it possibly it's impossible to tell, because I know that the duration of time that the mixing would have occurred is much shorter than the time it's lapses, but would you have any idea, was, I forgot my question, that's not good. Do you know, did they settle at any point? Was there any settling, or was it kind of one continuous, they moved to more Western Europe and then to the East?
Yeah, so we really have the only thing we really know is that we see this contribution in everybody outside Africa. The little contribution of Neanderthal see in Africa probably has to do with later migration from Europe to Africa, so indirectly bringing it there. You see this higher percentage in East Asia. Josh's work has sort of shown that there's at least 2 events of mixture then. But how this happened and sort of I was initially also very excited to see that on the X chromosome there is less contribution than on the autosomes, thinking this is mainly male contribution. But then we started seeing this part in the autosomes in the normal chromosomes too, where we had a reduction. So I don't dare say anymore that it's a mainly male contribution. But that's about everything we can say about socially or where it happened, I think.
This question here.
Hi, I was just wondering from the Neanderthal sequence how much of the soft phenotype of the Neanderthals can be reconstructed, things like eye color, hair color, anything of that kind.
Yeah.
That can't be inferred from the skeleton.
Yes. Of course, it is, as I said, in general, we are very bad at these things. There are these very depressing studies where we say easy phenotypes like at body height that we know is quite heritable. There are hundreds and hundreds of genes that contribute to that. So I would be very cautious. Even inferring things like eye color and skin color, you're sort of 70–80% correct in present-day populations when you do it, much less in the sort of Neanderthal background. You're sort of we can sort of sense that something there with skin and hair, but what, we really have no idea. In the future, perhaps.
Question here in the front.
Changing that now to the Denisovans, because we know much less about them, what can we reconstruct from the Denisovans and how they look like maybe?
Yeah, so I mean, people have looked at it and claimed they were brown-eyed and etc. I don't know how much, I suppose. So I would much rather hope for fossils. I mean, there are 2 teeth of the Denisovans that are very big and sort of look quite archaic, I understand from talking to the experts on this. I would very much hope that we will be able to retrieve DNA from some fossils, say in China or so, where one has morphology that we could show by DNA that they were Denisovans, so we could infer something about what they looked like. So.
Okay, Eamon, you're closest, and then Katerina, and then...
We've never found Neanderthals in Ireland. I'm just wondering, if we did find one, what would you expect to find in the DNA of an Irish Neanderthal?
That's a very good question.
Yeah, so it would be a redhead.
I was wondering whether, since you have now the symposium results, whether you think that Denisovans could be related to or could be Homo heidelbergensis.
Mm-hmm. Yeah, I think they could certainly be descendants of heidelbergensis. That's certainly possible, yes. That heidelbergensis might certainly be the common ancestor sort of Neanderthals and Denisovans. But the Denisovan in the Denisova cave is a lot later. It's even later, of course, than the Neanderthal in the cave. But we don't have a date really. What?
You could have late survival.
Maybe you had, yeah. Mm-hmm. Perhaps, yes.
Okay, last question down here.
In terms of understanding the hybridization and compatibility of different ancient humans, is there any way to tell the chromosome numbers from different
So the problem is we have these short fragments, of course. So we were very interested, for example, humans, of course, have 1 chromosome less than apes. Our chromosome 2 is a fusion of 2 chromosomes. So one idea was to say, wow, if there is an incompatibility, maybe it's a sort of, if you have different chromosome numbers, the hybrids generally have problems with fertility. So we looked for that junction fragment of chromosome 2 where they are fused, and both Denisovans and Neanderthals have that junction fragment. So they share this chromosome fusion. Now that said, if they had some other rearrangement that we didn't know to look for, if they had a fission of 2 chromosomes, we wouldn't know that. Because we really have to know what to look for in terms of this.
Okay, I'd like to thank Professor Pääbo one last time